So next up, we have a talk on a hot topic, gene therapy. Is it the new frontier? We've got Dr. Shawn McCandless here from Children's Hospital of Colorado. He is with the Anschutz Medical Campus there and is chair of the Department of Genetics and Metabolism. He's currently involved in a variety of clinical trials, including gene therapy trials for OTC. So let's welcome Dr. McCandless.
Thank you, Joe, and I'd like to thank all of the organizers. I just got to get my stopwatch so I don't go over, because I have a habit of doing that. Although, frankly, is it the new frontier? Yes, but the frontier has a habit of shifting, so thank you very much. You've been a great audience. Really, I think what I wanted, this is more going to be like Gene Therapy 101. And I thought we would start off with some definitions to make sure that the language I use later in the talk is everyone understands it. Many of you will be aware of what we call the central dogma of genetics, which is that your DNA is the code. When it's time to take that DNA and use it to make a protein, there's an intermediate step called a messenger RNA. So the DNA gets copied to make the messenger RNA. The messenger RNA has a lot less information in it. The DNA has all the information, not just how to make a protein, but how to turn a gene on and off at the right time and the right place, and to make just the right amount of the protein that you need in that cell. And genes can do other things. There are genes that only make RNAs, and there's all kinds of different things. But the messenger RNA has one job. That's the tightened version that is the instruction for how to make the protein. And so the process of converting DNA to RNA is called transcription, and translation is when that RNA is used to make a protein, as Dr. McLeod just talked about. So when we talk about gene therapy or nucleotide therapies, as I like to refer to them, because it can be either genes or messenger RNA or other kinds of RNA, we think about the vector, and to me the vector is the truck. That's what you put the load into, and the truck takes it somewhere and drops off its load. The payload is the gene or the mRNA or something else, and once that gene or mRNA the truck delivers that payload to the cell, it gets into the cell, and we'll look at a slide about this in a minute. That's when the DNA gets transcribed to make RNA and then translated to make a protein. And so the vector is the truck, the payload is either the gene or the RNA. And genes themselves can do a couple of different things when they get to the cell. Sometimes you can just drop off the DNA and it sits there, and when it's time to make protein, that piece of DNA gets made into a protein. And that's called a non-integrating situation. So the DNA, that gene therapy that you just delivered to the cell, is just kind of sitting in the cell by itself, just there waiting to be translated. And made into to have copies made, which are the messenger RNA, and then to be translated into a protein. There's other approaches where the gene can actually be integrated into your own DNA, into the person's own DNA. And there's some advantages of that and some disadvantages of that we're going to talk about. But the important thing about that is if the gene therapy gets integrated into the person's own DNA, it can then be copied and move. When the cell divides, the DNA gets copied and it goes into the new cells that are formed. Whereas a non-integrating gene therapy that sits in the cell, when that cell divides, that DNA is either going to be lost entirely, or it will only end up in one of the two cells that come from the cell division. And so when you have an integrating DNA, that gene therapy, that you're targeting to a specific place in the human genome, that's what people mean when they talk about gene editing. So that can be fixing a gene that's broken, or it can just be inserting a gene someplace else in the genome to do something. And again, that's often what we're trying to do, but sometimes it can be undesirable as well. And we'll talk about that in a minute. So just a few definitions. Payload is the gene, vector is the truck. I kind of use those terms interchangeably. The key to gene therapy is getting the gene therapy to the cells that need it. And in the urea cycle world, for most of the disorders, we're pretty fortunate, especially the earlier ones in the cycle, because they primarily need to be expressed in the liver. So you don't necessarily have to treat the rest of the body. And the major differences there would be argininosuccinic aciduria, ASA, or lyase deficiency, or arginase deficiency, where the enzyme is doing things besides just the urea cycle. And so fixing the liver will fix some of the problems, but not all of the problems for those conditions. The good news is that the liver is a common target for gene therapy, so there's been lots of research done in developing ways to get gene therapy into the liver effectively. It's one of the more well-studied tissue types. The eye and the bone marrow have some special advantages. The bone marrow, because you can take it out of the body, take it to the lab, and treat it in the lab, and then you can give the person their own bone marrow back as a bone marrow transplant, and you've now corrected their genetic defect. So for sickle cell disease, that's the approach that people are taking, and that's been very effective and has some real advantages. The eye is advantageous because you can inject the gene therapy directly into that small space in the eye and deliver a lot of the gene therapy to the place where it needs to go. Unfortunately, the liver is much bigger than the eye, and it's not so easy to deliver directly into the liver, although there are ways to do that. The brain has its own challenges because our brains are very well protected from things that get into our bloodstream to prevent them from getting into our brain. Only things that your brain wants to come into the brain cross what's called the blood-brain barrier. So if you just inject viruses into somebody's bloodstream, that's not going to go into their brain very efficiently. And so if you want to treat a condition in the brain, you need to inject the gene therapy directly into the brain. And then, even then, it's not always easy to know where the gene therapy is going to end up. So again, we're fortunate in the urea cycle disorders that we don't really need to worry about treating the brain. We really just need to focus on the liver. So I want to talk a little bit about the vectors, the trucks that we use, and you'll hear about different terms. All of them, except for the last category, are essentially types of viruses. And viruses are naturally occurring cells, organisms, that really are just there to copy themselves. They get into another host, and they get into the cells of that host, and then they just copy themselves over and over. That's what they have evolved to do. So some of the early studies in gene therapy used a particular kind of virus called adenovirus. And adenovirus is attractive because usually adenovirus infections aren't that bad. And it's kind of a big virus, so you can put big genes into it. You can put a lot of payload into that truck. It's like a massive truck. But unfortunately, adenoviruses cause a lot of inflammation. And so that has not worked out very well. And now there's been a lot of research in the last 30 years, 25 years on these adeno-associated viruses, which are smaller, which is a disadvantage, but they're much less likely to cause inflammation. They still cause inflammation, and they still have all of the problems of other viruses, or many of the problems of other viruses, but they're less, and they are easier to manipulate. And there's been so much work done that there's now lots of different sort of flavors of adenoviruses. And when I say flavors, what do I mean? What I mean is that outside of the virus, there's proteins that are attached to that, to what we call the capsule, or the membrane around the virus. And those proteins find something that they bind to on the human cells. And so if you get the right AAV that has the right proteins on the surface of it, it may be more likely to attach to liver cells than, say, muscle cells or blood cells or something else. So there's all kinds of different forms of AAV with different proteins on the surface that make it more or less likely to attach and deliver its payload to particular kinds of cells. And at the bottom of the slide, you can see that there's a list of different types of AAVs and what tissues they are most, the term we use is trophic, they're most likely to attach to. And AAV2 and AAV8 are both particularly trophic for the liver, but you can see that AAV9 also goes to liver. AAV2 binds to lots of different tissues, and that's one that's often used for muscle problems. Many of the genes that are targeting the liver for urea cycle disorder, the gene therapies, are using AAV8 right now. That could change in the future. Lentiviruses are more likely to be used for gene therapy on bone marrow transplant, where you take the cells out of the body. Lentiviruses and retroviruses are interesting in that the virus itself is able to insert the DNA and integrate it into the human genome. So lentiviruses, you don't need any additional packaging to incorporate the DNA into the human genome. The downside of a lentivirus is that when it does that, it's not always in the place that you want it, and that can cause problems, which we'll talk about in a minute. And then the last thing that and I'm not going to talk any more about lentiviruses because they're not really pertinent to our needs right now. The last thing I'm going to mention is this concept of nanoparticles. And nanoparticles are artificially made packages that are even less reactive, less likely to cause an immune response than the AAV vectors. And so those are, they're being developed. There's lots of different ways to make nanoparticles, and they may be in the future a more desirable delivery system, but they're not quite ready for prime time for gene therapy yet, although we will talk about them because some of the messenger RNA therapies are using them. So we've alluded to this, but how does gene therapy actually work? Well, in the simplest form, the vector, the truck on the far left, finds the cell, the proteins on the surface of the cell and the proteins on the surface of the vector are compatible, and so it binds, and then it enters through the membrane. Actually, the membrane takes in the virus, and then the virus particle attaches to the nucleus where the DNA lives, and it releases the product. Your body's machinery then takes over and makes a messenger RNA from that DNA, and then makes protein from the messenger RNA. The gene product is called a transgene, and in this example, it's not integrating into the DNA, it's just sitting there on its own, and so that works very well, and it's safe because it's not disrupting the DNA. Now, I said lentivirus is particularly concerning because I shouldn't say concerning, but lentivirus is known to incorporate its payload into the DNA. AAVs usually don't. And what do I mean by usually? Probably about 2 to 3% of the time they do. So there's always the possibility, even with an AAV vector, that there will be some integration into the human genome. I just want to take a second to say these treatments require a lot of trucks. 10 with a lot of zeros after it. A typical dose is going to be in the range of 1 times 10 to the 13th copies of the virus particle or the viral genome per kilogram. Maybe, and that's kind of on the low side of the dosing range that people are using. I think Dr. Grimm will talk maybe about that in a minute. Some of the muscle diseases are using 10 to the 14th or 10 to the 15th copies of viral genomes. That's a lot of virus. To put it into perspective, if somebody with COVID sneezes on you, that's going to be about 100,000 times less virus than you're going to get from your gene therapy injection. It turns out probably the more virus particles you get, the more likely you are to have side effects or complications from the treatment, although that's a complicated issue. So you've heard of CRISPR, which is a system that bacteria have evolved to protect themselves from being infected by viruses. And the CRISPR process has been engineered by scientists to allow editing of the human genome. And basically, without going into too much detail, which would only embarrass me by showing how little I understand it, the CRISPR has essentially two components. The CRISPR itself attaches to the DNA and breaks the DNA, and then there's a separate component that's a guide that in some cases can help the DNA to repair itself in a way that inserts the DNA that you're interested in, the sequence of the DNA that you're interested in. So you have to deliver both the CRISPR and the template, and so it makes it a little bit more complicated. But depending on how you design the CRISPR molecule, it can be very specific about where it inserts into the DNA. So you can actually use that to correct a specific genetic mutation. The problem is that everyone in this audience who has a urea cycle disorder probably has a different mutation, and it's unlikely that anybody's going to make a CRISPR molecule that's specific for your mutation. So what companies have been doing is creating CRISPRs that will insert the gene in a particular place in the genome, in a place that's going to be safe, but also going to be effective. So if you can use CRISPR to insert the gene into a piece of DNA that's going to be turned on in the liver all the time, then that piece you've just added, if it happens to be OTC or arginine-succinic acid synthase or some other gene that you're interested in, it will be turned on all the time and making lots of the protein that you're missing. So gene editing CRISPR is used for gene editing. And there are other things too. There's things called zinc fingers and talens, T-A-L-E-N, that also are used for gene editing. And they all have advantages and disadvantages. We talked a little bit about non-integrating versus integrating. And again, just to remind you that the non-integrating, it's good because it delivers the DNA, it doesn't get incorporated into the DNA, so you're less likely to have problems with risk of cancer and some of the other rare complications you may have heard about with gene therapies. But it has the disadvantage of as cells divide, it has the potential to then kind of taper and wane a little bit in effectiveness. And we don't know how long this is going to take in liver cells. Liver cells are kind of intermediate. Blood cells turn over all the time. And so if you did a non-integrating vector into blood cells, you would lose that very quickly. In the liver, the initial studies in humans seem to show that the effects are lasting for years, and we don't know how long that's going to be. The integrating vectors or techniques for gene editing have the advantage of when the cell divides, the DNA gets copied and goes into the daughter cells. And so there's the potential to keep this gene therapy going forever if it's integrated into the genome. And in fact, if fixing that gene in one cell gives it an advantage compared to other liver cells, over time, you may get even more and more cells in the liver that are corrected. So there's some potential advantages to that if we can figure out how to do it safely. And then the last thing we talked about is the messenger RNA therapy and how does that work. And on the screen, there's a whole bunch of different kinds of RNAs, and you may hear somebody talking about ASOs, or which are also called allele-specific oligos, and those are mRNAs that bind to a particular sequence of DNA or RNA, and they may enhance making a particular protein, or they may block making a particular protein. So if you have a genetic defect where it's turning on a gene that shouldn't be turned on, and that's what causes the problem, you could use an ASO to suppress that process. What I really want to show here, though, is that what we're thinking about are using messenger RNA. Instead of DNA, you put messenger RNA in the truck. The truck delivers it to the liver, injects the messenger RNA, protein gets made from that. The downside of that is that messenger RNA gets rapidly degraded in your body. I say that's a downside, it's also an advantage. If you get gene therapy, if there's a problem, you can't take it back. The messenger RNA will eventually degrade, and by eventually it's probably within 7 to 10 hours that the messenger RNA disappears. The protein that's made from that can last days or even weeks in some cases. So there are some advantages and disadvantages to messenger RNA. It may be that in the future, messenger RNA goes to work almost immediately when you inject it. So it may be that if a baby's born and they're very sick, maybe we're going to put them on messenger RNA therapy as a temporizing measure to try to fix their urea cycle and get it working again temporarily until we can do something more permanent, whether that's a liver transplant or a gene therapy or some other approach. So you can imagine that there are challenges to that, because your insurance company is not going to be very happy if we call them up and say, oh, we'd like you to spend a few $1 million on messenger RNA therapy before we spend a few $1 million on gene therapy. But, so far, that seems to be working out, and that's at least in America, we tend to have a way of figuring stuff like that out, so I'm optimistic for the future. There are some challenges to gene therapies. In particular, these AAV particles are naturally occurring. They exist in nature, and so some people have already got them. They've been infected by them. You probably didn't know you were infected by them because they don't really cause much in the way of illness, but your body may make antibodies against it. So if you've tried to enroll in a gene therapy trial, you may have been told that you're not eligible because you're already immune to the vector, to the AAV, to the truck that would carry the gene therapy. So that may be as much as 25% of people for AAV8. Depending on where you live, it could even be higher than that. I feel confident that we will be able to overcome this problem in the future. There are a number of drugs that that temporarily modulate your body's immune system in ways that we may be able to get past this, either by if you need to be retreated in the future, or if you're already immune to that AAV, maybe there's a different truck that we could put it in. Now, that's a problem with the FDA, because right now the FDA says this vector and this gene is a unique therapy. Even if the vector is the same, the truck's the same no matter what you put in it, the FDA sees that as a totally different drug. And so the companies have to go through the whole process of getting them approved. That, I don't know, hopefully that we'll figure that out too, but I'm not optimistic. Again, maybe Dr. Grimm will have some ideas about that. A couple of other things. When you get viruses in your body has an immune response, and there's different types of immune responses. There's one part that's called the innate, well, first off, you can have a rapid immune response because you've already got antibodies to that virus, and that will cause problems. It will actually break down the virus particle and make it not work for delivering gene therapy. B cells are a type of white blood cell that make antibodies, and so once you test somebody, they're not immune to AAV8, you give them gene therapy, they're developing antibodies, and then you can't reuse that vector, at least right now. You wouldn't be able to reuse that vector, so you couldn't treat them a second time with the same treatment, at least right now. There's another type of immune cells called T cells that are responsible for a more delayed inflammatory reaction. And sometimes T cells can kind of get ramped up over a few days or weeks, and that can actually start to break down the virus prior to its really doing its job for delivering the gene therapy. And so many of the studies that you'll be hearing about now, and probably when we actually have these things available for treatment, we'll be using steroids or maybe something else in the future to slow that process down to make sure that the gene therapy has the opportunity to do what it needs to do. And then there is another kind of immune response called the innate immune system, where there are cells that are just there waiting for, in your body, waiting for something foreign to get into your body, and they release a whole bunch of chemicals called cytokines that set up inflammation immediately, and that can also be a problem with gene therapy. I think I've already talked about the future, though. I think that either doing something to alter the immune system temporarily, or using a different vector, a different truck, or the use of lipid nanoparticles in the future will allow us to get past some of these problems with people who are already immune. Sometimes the inflammatory response can be very serious. This is rare, but it's been seen multiple times now in people getting gene therapy with whatever vectors you're using, but especially the AAVs. This is a rare but real complication. There's a particular sort of inflammatory response that causes blood cells to break down and to get microclots that can damage the kidney and other tissues. That's called thrombotic microangiopathy. And that, we now know to be on the lookout for that. And if you can catch it early, there are some management strategies. The really scary things are what you may have read about with one of the muscle diseases, where they use a really, really high dose of vector, and somebody had this, what they call a cytokine storm. That's that innate immune system just releasing this massive amount of inflammatory molecules all at once. And people can get very, very sick very, very quickly when that happens. Again, places that do gene therapy are aware of this, and most of us have plans in place for monitoring and treating that. I don't want to say that we've mastered it, but I think we're on the right track to being able to have this not be something that's going to prevent us from using gene therapies. A couple of other biological challenges are that it's not clear if you've got pre-existing liver disease, and by that I mean inflammatory liver disease from hepatitis, or there are other metabolic diseases where there's a lot more inflammation in the liver than what Dr. Burridge was describing in our urea cycle patients earlier today. It's not clear that whether that's going to impact the gene therapy and make it less likely to be effective, but also maybe more likely to contribute to the complication at the bottom, which is this theoretical risk of cancer. So AAV doesn't usually incorporate its payload into the DNA, the person's DNA, but it can, rarely. And if it happens to get in the wrong place in the DNA and disrupt a gene that's important for preventing cancer or limiting the growth of cells, you may actually get that cell turning into a cancer cell. Again, hypothetical with AAV vectors. I don't think this has been described in humans. It's been described in mice, but there's some differences in the mouse genome that make it a little bit more likely to occur in mice. So if you come across that in your reading, don't get too upset about that right now. But it is a reason that we're going to have to monitor for a really long time. Cost is always going to be a barrier, at least for the as far into the future as I can see. Current gene therapies are somewhere between $500,000 to treat eye diseases to over $2 million for some of the newer treatments for neurologic diseases. There may be market forces that will bring these prices down, but for rare diseases, that's always going to be a problem. And the other thing to be aware of is that if you have a urea cycle defect, gene therapy may not totally eliminate the need for scavenger therapy. And so you may end up with having to argue with your insurance company. So I want to just take the last minute or two and talk about what does it mean if you're in a clinical, what should you expect if you decide to do a gene therapy clinical trial? The first thing you should expect to travel somewhere. And you should expect a lot of screening tests, including testing to see whether you're already immune to the vector they're using, the AAV vector they're using. There's probably going to be some overnight hospital stays, which could be triggering for some of our patients who've really had a bad experience in the past. There is an IV infusion. For a clinical trial, and you're going to hear more about this from Dr. Berry, so I'm not going to go into it too much, but there's going to be tests that they need to do to show that the drug is safe and that the drug is effective. Then there will be lots of visits over the next year. And remember, if you're in a clinical trial, there's probably a 50% chance that you're going to get placebo. You're not going to get the active drug the first time, but you will get it the second time. And in most gene therapy trials, that's about 48 weeks into it. So you get a second infusion. You and your doctor who's doing the study with you will not know when you get the treatment. Nobody will know. Although if you get the treatment and it works, it should become apparent over the next year. And then you'll have regular visits again in the second year, and then you should expect to have ongoing visits for the next 8 to 10 years at least for the monitoring for those long-term effects. Once it's approved and it's a treatment that you just go to get, you should expect, just as like you would in the clinical trial, to be treated with steroids for a month or two after the infusion of gene therapy, at least for the foreseeable future. You may need a liver biopsy depending on the clinical trial, although I'm not aware of any right now that are requiring that. You do need to stay in close touch with the research team, and if you get any kind of illness or anything weird, you need to let them know. And just know that if we knew that the drug was safe and effective, if we knew the therapy was safe and effective, we wouldn't have to do the clinical trial. So there's going to be things that we don't know, that we can't expect, or rare things that are going to turn up, even if the treatment has been used safely in many other people. mRNA trials are very similar, except that you have to get retreated over and over. And so you have more frequent visits, probably every 2 weeks, to get the treatment and to check for safety and effectiveness. May eventually be able to switch to home therapy if you're on a mRNA trial, but we're not quite there yet. And we already talked about the advantages and disadvantages. Once these things are approved, it's going to be similar, but less visits. It won't be quite as big a deal to get the treatment. In fact, you might not even need to stay overnight. Insurance, prior authorization from your insurance company is probably going to be the biggest barrier for all of us. And then the other probability, although we don't know this for certain, but for many of the gene therapies that are currently available, the companies limit the centers that are trained and certified to deliver it. So you may have to go somewhere else to get your treatment and then follow up with your own doctors locally, or you may have to even go to another center that's closer to home but still not your home treatment center for the follow-up visits. There's a few urea cycle specific issues to just mention. The first is, how many cells do you need to correct to fix the problem in the liver? And the answer is, we don't really know. We don't really know how many cells get fixed. I've seen all kinds of estimates in the medical literature. But we also don't know whether there's some cells that it's more important to treat than others in the liver. We know that there are cells around the portal veins in the liver where the gene therapy, or where the urea cycle is most active. And then for argininosuccinic acid, if we're only treating the liver, that may not fix the problem. We may need to figure out how to get the gene into the endothelial cells of the blood vessels and other places where argininosuccinic acid lyase is active. And then just to say that, again, there is potential to treat with messenger RNA if one of those gets approved until a person's ready for gene therapy. So I just want to leave you with some expectations. Gene therapy is not a cure. This is not going to be a cure, at least not for the foreseeable future. But the hope is that it will make life better, and that reduce the amount of treatment that you need and reduce the dietary needs, maybe eliminating them for some patients, but probably not for everyone. It's going to work better for some people than others for reasons that we don't fully understand. And then I do think that there's an idea that gene therapy is one and done. It's probably not, although right now it is. You wouldn't be, if you got AAV8 with a gene therapy, you probably wouldn't be able to get that again. But I think in the future, scientists will and engineers will solve that problem. The other thing to point out is that if you do gene therapy and it's not as effective as it needs to be, it doesn't preclude you from getting a liver transplant. So you can get a liver transplant after gene therapy if that's necessary. But what you probably won't be able to do is to be part of another gene therapy trial. No company is going to want to take the risk of having somebody who's already had gene therapy that might have a complication from the first trial they were in be in their study, and because then you couldn't sort out which, if you had a complication, you couldn't sort out which one was causing it. The last thing, which is the first line on the slide, is that you have to remember that neither the company that's making this treatment, nor your doctor who's giving you the treatment, nor your doctor who usually takes care of you knows how to treat somebody who's had gene therapy, somebody who was at this place on the spectrum of disease and now is moving to a different place in the spectrum of possibilities for that disease. So we're learning as we go with these gene therapy trials and trying to better understand how to take care of patients that get the therapies. And so I'll just leave you with this famous Winston Churchill quote that this isn't the end. It's not even the beginning of the end, but maybe it's perhaps the end of the beginning. And a lot of work yet to do, but man, I've been in this field for 30 years, and I've never been more hopeful or excited for the future than I am today. So thank you for your attention.